Okay, this part of the lesson is very easy because it's just descriptive, no major problem. So, B-cell receptor and antibody. What are the structures, the feature of B-cell receptor? Because then we will discuss a few molecular details which are very important. The receptor for antigen expressed by B-cell on the cell membrane is called BCR or B-cell receptor. Is a special immunoglobulin with a transmembrane domain and a short intracytoplasmatic tail. Okay, this is the first message of today, the important message. We already saw these pictures, the different structures of this picture. All the BCR on the cell membrane of a B cell have the same antigen specificity. Please, I hope you will not make a mistake when you will get this question as a test, because I will commit apoptosis if you are making the mistake. You can, so one lymphocyte, one T lymphocyte, one B lymphocyte, one specificity. All the B cell receptor have the same specificity on the surface of a single B cell. All TCR have the same specificity on the surface of a T cell. So all have the same binding site, even if IgD or IgM. So you cannot make a mistake in this question of the test, please. The BCR expressed by B1 B cell and B2 marginal zone B cell is made by monomeric IgM only. The BCR of follicular B2 cell are both monomeric IgM and IgD. All IgM and IgD with the same antigen specificity. So they have the same ability to bind, to interact with the same antigen. Okay, please, absolutely, you will have in your test a question about that. You cannot make a mistake. It's very important. All this interacts exactly here. They're exactly the same sequence of amino acids. They interact with the same target, IgM, IgD, when are on the membrane of the same B cell. And all TCR on the membrane of a T lymphocyte has the same specificity. So in follicular B2 cell, we have two kinds of support of the same binding site. So they're just a little bit different, IgM and IgD, but they have exactly the same target. Okay? Silvio is not convinced. No. The BCR expresses on the membrane and the Ig secreted by activated differentiated B cell in plasma cell are symmetric molecules made by 3 globular fragments. And I will show you that. So you can see the molecule in a globular way, like me. One part is called Fc, and the other fragment A and B. So the BCR and immunoglobulin bind the target antigen through a special region, I designed in purple, which is a binding site, the area in which the sequence of amino acids are different one to the other one. Initially, when the immunology discovered the immunoglobulin 100 years ago, they were believing it was a fragment A, a fragment B, a fragment C. and this was the name. Then what discovers is this fragment, this part of the molecule, and this part of the molecule are identical. So we have this kind of symmetric molecule made by two fragments, which are the one antigen binding fragment, and another fragment called fragment C. The third fragment, fragment C, mediates the biological activity of the immunoglobulin. So here is the part which is able to bind the target, and here will perform some biological activity. So this is why I mentioned asymmetrical molecule in three parts. So this is fragment C, the part COH terminal of the molecule. This is NH2 terminal. And we have one chain going in this way, which is a heavy chain, and another chain which is smaller, which is called light chain. So BCR and antibody are made by two chains, replica, symmetrical replica, so two heavy chain and two light chain connected by disulfide bridge. We connect all the molecules. The three globular fragments of the BCR and antibody are made by four proteins, two identical heavy chains and two identical light chains. Okay. And you understand what we can call fragment AB, fragment AB, fragment C. These two fragments are, these two parts, these two regions of the molecule are absolutely identical. Okay. And here is a binding site. With the antigen interact with the sequence of amino acid in this area, in the heavy chain and the light chain. This makes different every B cell from the other one. The sequence of amino acid in this part, the sequence of amino acid, and consequently the structural formation of this part. This is the binding site where the antigen may interact. This region is the region which interacts with the antigen, the binding site of the immunoglobulin. So the immunoglobulins, normally the monomeric immunoglobulin, this is a monomeric immunoglobulin, bind two different antigens, not two different, two antigens, which are the same in one part and the other part. So can bind two different parts of the same antigen, located in different parts in the space. Okay. These are glycoproteins because they have some sugar linked. This, if there was a BCR, you can add here the transmembrane domain and the cytoplasmatic tail. And here is a molecular structure of an immunoglobulin. You can see immediately that this molecule is made exactly, not exactly, similarly of what we saw with histocompatibility molecule of class 1 or class 2. There is always a replica of the same unit of almost 100 amino acids and a disulfide bridge. So seems several replicas of the beta-2 microglobulin. So you can see here 100 amino acid, here 100 amino acid. Each one is different. The sequence here is different from this one because they changed a little bit the gene. But you have 1, 2, 3, 4, 5 replicas of the same. And here you have 2 replicas of this, not of the same, of the similar stuff. Here we have the variable part. In blue is called light chain. In red, the heavy chain. And these are called the domain, constant domain 4, constant domain number 3, constant domain number of the heavy chain number 2, constant domain of the heavy chain 1, and the variable domain where change the sequence of amino acid because the ability to bind different kind of antigen. And this is a region called hinge region because hinge is this one, cartine, which allows the flexibility of the molecule. The immunoglobulins are molecules very flexible in the space because they don't know. You see, I designed this kind of symbol. They move, they are very flexible thanks to the amino acid, the peculiar amino acid located in the hinge region, which allow spatial orientation of the molecule. The globular structure of immunoglobulin rests on intrachain cysteine bridges here and interchain bridges which connect the various heavy and light chain. The heavy and light chain are linked by interchain disulfide bridges, cysteine. As I told you, this is a highly flexible molecule because the spatial amino acid sequence in the hinge region, which allow a major flexibility of this molecule. Something you don't have and is not required for the TCR because TCR doesn't need any flexibility or very minor flexibility. Okay? Okay, this is just to repeat something we already saw. The heavy chain are made by multiple of the basic molecular structure of about 110 amino acid with a disulfide bridge, 99, 100, 110. This is a basic brick by which almost many molecules of the immune system are made. We already saw the TCR, the class 1, class 2 immunoglobulin are made by using variation of the gene coding with this simple unit. Because this is a molecular economy, this basic globular, solid, resistant, flexible, and modular structure is exploited by the immune system to build several distinct molecules. Okay, you see here that is nothing else or something similar to various replica of the same molecule, variously organized. Replica of the basic structure may form long chains. Molecules made by exploiting this structure are considered member of the family of immunoglobulin superfamily. So we have many molecules. You will see many molecules in this course and in other courses where are made with this structure. These are member of the immunoglobulin superfamily. What does it mean? That are using the same brick to build many different molecules. And I guess I have not. Well, there you will find a picture of many different molecules made in this way. Sometimes very long molecules, various kind of receptors are made in this way. So a major B2 cell express about 10 to the 4 BCR, all with the same binding site. This BCR is made by immunoglobulin M and D. You already saw that. Monomeric IgM and IgD on the same membrane of the major virgin 4B2 cell share the same antigen binding site, but are different. What is the difference? The sequence of amino acid here in the purple chain is different from the one I designed in red. But also, IgD had 4 domains. The IgM had 5 domains, so it's longer. They produce more outside of the cell. So it's more flexible, the IgM with 5 domains instead of IgD with 4 domains. When this become antibody, they will be secreted without this part of the molecule. Okay, this R receptor, this is a BCR. You remember the TCR, T lymphocyte. TCR is made by something like that, you remember? Just like that, but was also unable to transduce the signal. To transduce the signal, the TCR have around the molecule, the zeta chain, and the gamma chain, the delta chain, they form the CD3 transducing signal. Here we have the same. This molecule have a tail, cytoplasmatic tail, which cannot transduce the signal. So to transduce the signal when immunoglobulin interact with the antigen, the signal is reduced when they do interact. The signal is transduced by a peculiar kind of molecule, which are located here on the basis, which are called Ig alpha and Ig beta, which possess the famous item region, the one which can be easily phosphorylated upon activation. And when the antigen interact, they perceive the modification of the immunoglobulin, they become phosphorylated and act as docking site to different kinases, which then transduce to the nucleus and activate the gene. They possess the item region. Ig alpha, Ig beta, monomeric. My Ig alpha are identical to yours, and to yours Ig alpha and Ig beta are identical in all of us. These are just to transduce the signal. They are member also of the superfamily of immunoglobulin, has the same basic structure. Signal transduction is done by Ig alpha, Ig beta, transmembrane protein of Ig superfamily with item motif in their cytoplasmatic tail. Item immunoreceptor tyrosine-based activation motif. Item is a conserved sequence of 4 amino acids that are repeated twice in the cytoplasmatic tails of certain surface protein of the immune system. Item repeats are separated by this, not important. So here are the region which can easily phosphorylate. The tyrosine become phosphorylated following protein modification. The phosphorylated tyrosine forms the docking site of other protein involved in the signal transduction. Exactly as we saw with the CD3 and the zeta chain of the TCR. So once the immunoglobulin interact with the antigen, they perceive the interaction, they become phosphorylated, and this becomes the docking site for the transduction of the signal. So when we have the activation of a B lymphocyte, the series of events we will see very soon are similar to the one we described yesterday for the activation of a T cell. The BCR, which interact with an antigen, change morphology, change conformation, move to the lipid raft, the area where there are a lot of kinases in the B cell are different. There is the same phosphatases, the CD45, and you will see we have the activation of the signal and the transduction of the signal through the phosphorylation of the item located on Ig alpha and Ig beta in a way very similar to the what we described for T cell.